Welcome to the Deep Dive. Today we are strapping in for a journey that goes way beyond just managing weight or blood sugar. We're looking at a stack of really cutting-edge research that asks a pretty revolutionary question.
Yeah, really is.
Are these new metabolic therapies, the GLP-1 receptor agonists and, a few other surprising interventions, are they actually capable of slowing down or even reversing the core biological process of aging itself?
That is the core mission today. Yeah, we're going to synthesize findings across, everything. Metabolism, cardiology, neuroscience, and deep gerontology.
Right.
We need to cut through all the noise around drugs like semaglutide and tirzepatide to really get at their pleiotropic benefits.
Okay, what do we mean by that exactly?
It just means their effects that go way beyond their primary purpose. And then we'll see how those stack up against some really novel anti-aging strategies. Things like metformin, hyperbaric oxygen therapy, and even some of the riskier frontiers like plasmapheresis.
And the source material we have here is, it's incredible in its breadth. We're talking about multi-omic deep dives tracking gene expression across, entire mouse bodies.
Yeah, the whole organism.
Then we've got real-world data comparing cardiovascular risks in hundreds of thousands of patients, and even human studies using, the gold standard biomarkers of biological time, the epigenetic clocks.
It's a comprehensive look. This deep dive is really about what science thinks might be the future of extending our health span.
So let's jump right in with the agents that have just completely captured the public imagination.
Yeah.
The GLP-1 receptor agonists, or GLP-1RAs.
Right.
So for the record, they're approved for type 2 diabetes and for weight management. They're typically given as a weekly shot with semaglutide doses going from, say, 0.25 milligrams up to the 2.4 milligram weight loss dose. But the whole story, the narrative around them, it has shifted so dramatically. It's no longer just a drug for diabetes or obesity. It really seems to be an all-purpose systemic modulator.
And that shift is happening for a really good reason. It's because of a fundamental biological discovery, just how widespread the GLP-1 receptors are.
Oh, right.
People initially thought, they were mostly in the gut and the pancreas just to regulate insulin.
That was the original thinking.
That was it. But now we know those receptors are, they're all over the body, in the kidney, the lung, the heart, and maybe most critically for longevity, the brain.
The brain.
This widespread signaling system is really the basis for all those whole body benefits we're starting to see.
And once you trace that metabolic thread right into the central nervous system, you immediately hit these aging hotspots.
Absolutely. The evidence linking poor metabolic health and accelerated brain aging is it's staggering. The sources show that having type 2 diabetes, it doesn't just increase your risk for certain things, it effectively doubles your risk for Alzheimer's disease.
Hold it. Wow.
It's a massive acceleration of neurodegeneration. So that creates this really compelling scientific mandate. If you can treat the metabolic dysfunction, maybe you can protect the brain.
So the scientific rationale is definitely there. But what about the actual molecular findings from the lab research, from the preclinical stuff with semaglutide? What are they seeing?
They are seeing profound neuroprotective effects. In the lab, these GLP-1 agonists, they seem to act like a general maintenance crew for your neurons.
A maintenance crew, I like that.
Yeah, they reduce neuroinflammation, they mitigate oxidative stress from free radicals, they decrease programmed cell death, what we call apoptosis, and they even promote the formation of new neurons. Neurogenesis.
So it's not just one thing.
No, it is the definition of multi-pronged protection.
It almost sounds like they're fighting the hallmarks of Alzheimer's and Parkinson's at the same time.
They are, and the data gets really specific. In models that are designed to mimic Alzheimer's, semaglutide reduced the typical hallmarks you see, like the A plaques and the neurofibrillary tangles.
Okay.
And in models for Parkinson's, the compound protected neurons against the specific toxins that researchers use to cause that PD-like damage.
You can actually measure this.
You can. To make it tangible, one source detailed that in obese mice, semaglutide treatment improved their learning and memory. You could see it in a shorter platform latency in the Morris-Water maze assay.
So they were literally cognitively better.
They were, because their metabolism was better managed.
That measurable improvement is so key. Now let's connect this to the whole body, because we have data from another one, exenatide, that suggests it acts as a true systemic anti-aging agent.
And that's where the multi-omic analysis comes in. It's like conducting a full-body diagnostic on an aged organism.
Right.
Researchers did this study on aged mice treated with exenatide. They looked at the transcriptome.
Which is basically the body's instruction manual, right? The RNA.
Exactly, the whole set of RNA molecules.
Mhm.
And they found the drug systematically counteracted age-related changes in that manual across 7 key organs and tissues.
7 organs. That sounds less like it's hitting one switch and more like it's hitting the master control panel for aging.
That's a great way to put it.
So which tissue stood out?
The effects were incredibly robust in the central regulatory tissues, so the hypothalamus and the frontal cortex in the brain, but also in high-demand tissues like the heart, skeletal muscle, even circulating white blood cells.
And what's the common thread there?
The common thread is the systemic dysregulation that comes with aging. This drug class seems to tackle multiple aging hallmarks at once. It fixes deregulated nutrient sensing, it reduces chronic systemic inflammation, inflammaging, and it dampens oxidative stress across all these different organs.
And this wasn't just, lines on a chart. It actually translated into physical improvements, didn't it?
Absolutely. The treated aged mice, they showed better forelimb grip strength and enhanced performance on the rotarod test.
Okay.
And crucially, these anti-aging gene expression changes and the functional improvements, they were specific to the aged mice. You didn't see them in young adults.
Ah, that's interesting.
Which strongly suggests that the GLP-1RAs are restoring function that was lost to aging, not just boosting an already healthy system.
That molecular data paints an amazing picture of systemic protection. But let's bring it to the highest stakes arena for humans.
Yeah.
Reducing cardiovascular risk.
Right.
Because now we have both semaglutide, the single GLP-1 agonist, and tirzepatide, the newer dual GIP-GLP-1 agonist. So there's this massive push for real-world head-to-head comparisons.
And this raises a really important question for doctors and for patients. Does adding that GIP component, making it a dual agonist, give you a distinct advantage in protecting the heart compared to a single agonist that's already highly effective?
We have a large database study on this. It compares people starting these treatments in just routine clinical practice. And it's fascinating how the real-world data seems to mirror the earlier controlled trials.
It really does. So the first comparison just confirmed what we already knew. Semaglutide versus a lesser proxy, sitagliptin, it successfully lowered the 1-year risk of heart attack or stroke. The data showed a hazard ratio of 0.82, so a significant benefit.
Okay, so semaglutide is a proven winner for the heart. No surprise there. But let's get to the real main event, tirzepatide versus semaglutide. Did the dual action prove to be superior?
And this is the surprise? No.
No.
For the primary composite outcome, which was myocardial infarction, stroke, or all-cause mortality, the results showed comparable benefits. The hazard ratio was 1.06, which, statistically means there was no significant difference between them.
Wait, so even with the dual mechanism, you're not getting more cardiovascular protection? You'd think 2 targets would be better than 1.
That is a great question, and it likely comes down to receptor distribution. While the GLP-1 receptors are abundant all through the cardiovascular system, GIP receptors are actually pretty scarce there. So tirzepatide's main advantage, the enhanced glucose control and maybe better weight loss from the GIP part, that doesn't necessarily give you a huge extra benefit in direct heart tissue protection compared to the already powerful effects of GLP-1 signaling alone.
So they're both good.
For reducing major adverse events, the benefits for both of these modern therapies are comparable.
Was there any separation at all between them? Any area where one did pull ahead?
A subtle one, yeah. When researchers looked at a secondary endpoint involving heart failure, so hospitalization for it, urgent visits, or all-cause mortality, tirzepatide showed a modest trend toward a benefit.
How modest?
An HR of A0.91. So it suggests maybe a slight edge in managing heart failure risk, but, overall, for general cardiovascular event reduction, they really do stand toe-to-toe.
Okay, so we've established the systemic protective power of these new GLP-1RAs. Now I want to pivot and look at some very different longevity contenders, starting with a drug that's been treating diabetes for decades, metformin. Why does this old standby continue to be discussed as a potential, anti-aging blockbuster?
Metformin is just fascinating because its effectiveness against aging was discovered almost by accident. It targets several of these fundamental pillars of aging that were first identified in, like, worms and flies. It works by inhibiting mTOR, which is a major nutrient sensing pathway, the same one targeted by calorie restriction. It also decreases insulin signaling and reduces the generation of reactive oxygen species.
So it's hitting that nutrient sensing pathway, kind of mimicking the benefits of chronic fasting, but without the actual fasting.
Exactly. And the data supporting its impact on human longevity is starting to look very convincing, especially from these observational studies that try to mimic randomized trials.
Let's talk about one of those.
Okay, so this one source used a really rigorous method called a target trial emulation study. They looked at women aged 60 and older with type 2 diabetes and compared those starting metformin versus those starting a sulfonylurea. And they specifically tracked for exceptional longevity.
Which they defined as what?
Survival to age 90 or older.
And what did they find?
They found a truly robust signal. Metformin initiation was associated with a striking 30% lower adjusted risk of death before age 90. That gave it a hazard ratio of 0.70.
30%. That is a huge potential benefit.
It's huge.
Now, you mentioned a crucial caveat about this kind of data. We have to be really clear about that.
We absolutely do. The caveat is that this was an observational study comparing two active treatments. It wasn't metformin against a placebo in a randomized trial.
So the groups might be different from the start.
Exactly. People prescribed one drug might be inherently different from people prescribed another. Now, the researchers used very sophisticated methods to adjust for that, but you can't definitively infer causality.
But the signal is strong.
The signal is strong, and the consistency across multiple studies strongly supports metformin's role as a potent modulator of aging in humans.
Okay, let's move from a molecule to a physical intervention. Hyperbaric Oxygen Therapy, or HBOT. This involves breathing high-pressure oxygen. How on earth does this relate to fighting biological aging? It seems kind of counterintuitive.
It is, but this is where it gets really interesting. HBOT relies on what researchers call the hypoxic paradox. By intermittently exposing the body to 100% oxygen under high pressure and then following that with normal air, you're creating a controlled adaptive stress response.
So we're not just saturating the body with oxygen, we're intentionally causing this fluctuation, almost like a controlled shock, to get a beneficial response.
That's the paradox. This intermittent hyperoxia, it triggers cell survival mechanisms, and it activates these key transcription factors like HIF1 and NRF2. They're the master switches for cell repair and homeostasis.
So you're tricking the body.
You're tricking the body into thinking it's under stress, and that leads to these really robust protective changes.
What actual measures of aging does it impact? What can we see changing?
The results in human studies are profound. HBOT has been shown to increase telomere length.
The little caps on our chromosome.
The protective caps, exactly. And it's been shown to decrease the concentration of senescent cells.
The zombie cells.
The zombie cells that accumulate and drive inflammation as we age. So by clearing those out and promoting stem cell proliferation and neurogenesis, HBOT is really attacking aging from several distinct fronts.
So we've talked about taking powerful molecules using high-pressure oxygen. Now let's discuss removing something entirely. Circulating plasma factors. This brings us to plasmapheresis.
Right.
The idea of just removing the bad stuff that builds up in our blood plasma over time. What was the rationale behind the human study we looked at?
The rationale really came from some famous animal research suggesting that if you do a plasma exchange, you could rejuvenate older tissues by removing these pro-aging factors like certain inflammatory cytokines.
Okay.
So this human study took healthy blood donors and used a protocol of 8 plasmapheresis sessions over 18 weeks. They wanted to test its effects on standard clinical markers and, crucially, on the deep biological markers of age.
Let's start with the basics. What were the immediate clinical benefits of just removing plasma?
Well, the protocol was successful in what it was expected to do. Participants saw significantly lower levels of total cholesterol, non-HDL cholesterol, and triglycerides, all key markers for cardiovascular risk.
So on paper, it looked like they were cleaning up the system.
Exactly. They also saw a decrease in total proteins and albumin. Clinically, it looked good.
But the real surprise, and this is the major warning in this research, came when they looked at the core biological age markers. Before we get to those results, we have to explain what we mean by epigenetic clocks.
That's essential, yeah. When we talk about age, we usually mean chronological age, how many years you've lived.
Right.
But these epigenetic clocks, like DNM grimmage or the Dunedin pace of aging, they don't measure years. They measure your biological age by tracking methylation patterns on your DNA.
So it's like reading the accumulated wear and tear on your cells.
That's a perfect analogy, the software updates on your cells. And these markers are powerfully correlated with your future risk of disease and mortality.
Which is why seeing a change in them is such a big deal. So what happened? What did the epigenetic analysis show after the plasmapheresis?
Well, despite that initial clinical cleanup, the chosen protocol utterly failed to show any epigenetic rejuvenation.
It failed.
It did worse than fail. Instead of rolling back the clocks, the repeated plasmapheresis was associated with an increase in aging markers. The DNAM-grimage and the Dunen pace of aging markers both suggested that the subjects had biologically aged faster during the protocol.
Wait, hold on. So they pulled out the bad cholesterol, but they made the patient biologically older. How can removing pro-aging factors lead to accelerated aging?
And that is the central cautionary tale of this source material. The researchers believe this outcome was highly dependent on the procedural specifics.
Gotcha.
Repeated plasma removal, when it's done without the careful replacement of beneficial components like fresh albumin or other protective factors, it likely caused an acute physiological stress response. And this stress, or maybe the sudden loss of important protective components in the plasma, led to a negative biological alteration that is consistent with accelerated aging. It just shows how sensitive these interventions are.
So it's not enough to just pull out the old stuff.
No, you have to be incredibly careful you don't remove beneficial things or just induce shock to the system.
Okay, so to synthesize this incredible landscape for you listening, we've moved so far beyond simple weight management.
We really have. We have compelling multi-omic evidence that existing metabolic drugs like the GLP-1RAs, they profoundly modulate the systemic underpinnings of aging, from inflammation, neuroprotection, all the way to dramatic cardiovascular risk reduction.
Right. And metformin shows this powerful link to exceptional longevity by targeting nutrient sensing.
And then interventions like HBOT show promise by, forcing cellular resilience through controlled stress.
Okay, so let's unpack the aha moment here. The key takeaway isn't just that these treatments work, it's how they seem to work. The most promising strategies, whether it's targeting metabolism with GLP-1s or oxygen with HBOT, they all seem to work by forcing a systemic recalibration of our fundamental biological pathways.
That's it.
They make the body's internal systems behave as if they were younger, rather than just replacing or removing components, which, as we saw with plasmapheresis, can actually backfire in a dramatic way.
Precisely. The research just overwhelmingly confirms this inseparable link between your metabolic health and your cognitive and vascular longevity. And while the clinical benefits of GLP-1RAs for diabetes and obesity are clear, the big scientific question is still out there.
Which is?
Which combination of these molecular pathways, nutrient sensing, inflammation, telomere maintenance, stem cell health, which ones will the next generation of clinical trials need to target and validate to confirm an actual, measurable, and sustained reversal of human biological age?
That is the question for the future. And you now have the knowledge to track those developments with a really informed eye.